Good afternoon. My name is Don Gwan-an, and I'm PhD student in mechanical engineering at Hanyang University in Korea. The topic I will be presenting today is the development of a hemodynamic index for the carotid atherosclerosis using 4D flow MRI. Atherosclerosis of the carotid artery is a very dangerous disease with a high possibility of leading to stroke. In addition, the influence continues to rise as life expectancy increases. Diagnosis is usually based on how much the carotid is blocked to determine how to treat it. However, there are some exceptions that cannot be checked in this way. Because of this, there is an increasing number of hemodynamic studies that seek to analyze disease from the internal blood flow, away from the morphology. However, although many hemodynamic parameters and association with the disease has been reported, only a very few cases have been used for actual diagnosis. This is because it requires a high degree of expertise in both fluid mechanics and medicine. Therefore, for the practical application of hemodynamic studies, a simplified quantitative criterion that integrates all of these hemodynamic parameters is required. The 4D flow MRI is a visualization technique that is widely used in hemodynamic study. It has the advantage of being able to non-invasively measure a fake target, such as internal blood flow, using commercial MRI. So we use this technique for our research objective. So we set our research objective as to define a simplified hemodynamic index by integrating hemodynamic variables and comparing it with the conventional angiography-based analysis method. To achieve this objective, the following process was carried out. We acquired the 4D flow MRI data using systemically categorized carotid artery phantoms, and we calculate the hemodynamic variables from the velocity data. And also we perform the non-dimensionalization and normalization for quantitative comparison. And we define the hemodynamic index using normalized and dimensionless hemodynamic variables. And finally, we validate HDI by comparison with the traditional angiograph-based analysis. So I will present this brief explanation for this process today. First, for this study, we select 8 patients who underwent endarterectomy for atherosclerosis. These 8 patients was classified by the closure method. The 4 patients were patchily filled, and the other 4 were primary closures. The 3 types of carotid artery models were selected from each patient. First one is contralateral carotid, what we call control, and second one is a free operative carotid, which we call free, and last one is a post-operative carotid, which means carotid artery after 1 week of carotid endarterectomy. So total 24 carotid models were classified, and using the CT image of these 24 carotid arteries, the real-sized and rigid body phantoms were created with a 3D printer. Next, an experimental device was constructed using real-time controlled pump and flow meters. And actual perceptive flow conditions were implemented based on the ultrasound Doppler that obtained from each patient. As the working fluid, an aqueous solution of glycerin was used to characterize the viscosity of blood. and a small amount of copper sulfate was included in working fluid to enhance the MRI signal. For 4D flow MRI, Bruker 4.7 Tesla animal MRI machine in KBSI Ochang Center was used, and the spatial and temporal resolution was set isotropically to 350 micrometer and 25 milliseconds to measure the data. So using our configured experimental setup, we measured the 4D flow MRI data for these 24 carotid artery models as follows. The obtained data was segmented into an inner fluid lesion and the outer noise lesion using the OFS method. And based on this, the location of wall surface was established in proper way. Also, to reduce the noise effect, a divergence-free smoothing filter with an wall-bounded condition was applied to the velocity data with the house code. And this is the measured 4D velocity field that visualized for the model highlighted in red boxes. Next, key hemodynamic variables from the velocity field were calculated, which has been reported to be associated with the disease in other studies. For example, the flow rate, and the second one is washer stress and OSI, which is very popular to this academic area. And the pressure field, which calculated using the Darcy's law, and the recirculation zone, which means the region that flow opposite side to the mainstream directions. And next one is a stasis, which means the low speed region. And last one is an abnormal region, such as a stenosis prone and high shear region. And this abnormal region is reported to be associated with atherosclerosis in our previous study. And since the calculated variables have a different dimension, non-dimensionalization was performed for quantitative comparison. Non-dimensionalization was performed based on Buckingham pi theorem, and the blood density, the hydraulic diameter at common carotid artery, and the mean velocity at the same lesion were used as repeating variables for mass, length, and time dimension respectively. Next, we normalize the non-dimensionalized variables. To quantitatively compare variables of different scales, we first scaled each variable to have a value between 0 to 10. Then some variables were transformed so that the value of the variables also increased as the degree of stenosis increased. The degree of stenosis is a measure of how much the carotid artery is blocked and was defined as shown in the lower right figure and used as a reference for our non-dimensionalization and normalized variables. With this normalized and dimensionless variable, the HDI was defined using a weighted sum method. The weights for each phi used as defined as following using the R-square value obtained from the linear regression of the phi for the degree of stenosis. This is the HDI result for each carotid artery model. If the HDI is close to 10, it means it is more likely to develop problems with severe stenosis. And conversely, if the HDI is close to 0, it means that there is a high possibility of problems caused by overextension. To verify the validity of HDI, we define the angio-based index using a traditional method. And also we compare and analyze this value with our HDI. You can see that the value of the angio-based index are discretely distributed for each group of pre and control and forced cases. On the other hand, the HDI can confirm that the value for the three groups are continuously distributed compared to the angio-based index. This means that HDI allows more detailed analysis than conventional method. For example, the models highlighted in the red boxes have an angio-based index close to 5, but the HDI is close to 7. This can be interpreted as the models having no problem from the morphological point of view. However, in the hemodynamic point of view, there are some factors that promoted stenosis in the internal flow. Similarly, that HDI shows that the model highlighted in the blue boxes are expected to have adverse effect due to overextension from a morphological point of view, but are closer to normal from a hemodynamic point of view of internal flow. From this result, we can confirm that the use of the defined HDI has the potential for more efficient and precise diagnosis than conventional morphological method. However, our HDI still needs a lot of improvement. Therefore, as future work, we intend to use patient's follow-up and medical data with the clinical opinions obtained from specialists to improve the mathematical definition of HDI. And also we present clinical guideline for HDI value, and it is our final objective for our research. And this is our summary of the presentation, and thank you for listening.